For millions of years, mammals have enjoyed a remarkable form of protection against fungal infections, our warm bodies. This thermal barrier has limited fungal pathogens capable of infecting humans to just a few hundred out of millions of species. But as our planet warms, this ancient protection is beginning to crumble. Join us as we explore how rising global temperatures are driving rapid thermal adaptation in fungi, creating an unprecedented challenge that could reshape our understanding of infectious disease in the 21st century. For millions of years, mammals have possessed a remarkable defense mechanism against fungal infections, our warm body temperatures of 36 to 37 degrees Celsius. This thermal barrier has been an evolutionary masterpiece, effectively preventing most environmental fungi from colonizing our bodies. Out of the estimated 2.2 to 3.8 million fungal species on Earth, historically only about 300 to 400 have been documented to cause human disease. This protection mechanism is believed to have evolved during the mass extinction event at the end of the Cretaceous period, when mammals' higher body temperatures may have provided critical protection against environmental fungi that devastated cold-blooded reptilian species. In essence, our warm bodies have been our first line of defense against a kingdom of potential pathogens that vastly outnumbers the bacteria and viruses we commonly fear. Recent research published in the Proceedings of the National Academy of Sciences has revealed a disturbing trend. Rising global temperatures are driving rapid thermal adaptation in fungal species, enabling them to tolerate higher temperatures that were previously lethal. Researchers at Duke University demonstrated that particular Cryptococcus species, when exposed to gradually increasing temperatures in laboratory settings, can adapt to survive at 37 degrees Celsius in just 800 generations, potentially just decades in natural settings. This thermal adaptation occurs through genetic changes that alter membrane fluidity and heat shock protein expression, allowing fungi to maintain cellular integrity at temperatures that typically cause protein denaturation and cell death. Dr. Aaron Mordecai of Stanford University has documented that the optimal growth temperature for hundreds of fungal species has increased by an average of 0.4 degrees Celsius since the 1960s, closely tracking global temperature increases. This adaptation appears to be accelerating, with fungal thermal tolerance shifting more rapidly in the past decade than in previous periods. The mechanisms behind fungal thermal adaptation are complex and multifaceted. Laboratory experiments at the Weizmann Institute of Science have demonstrated that when subjected to thermal stress, certain fungi can activate transposable genetic elements, jumping genes, that increase mutation rates specifically in genes related to temperature tolerance, effectively accelerating their evolution in response to warming conditions. These adaptations involve sophisticated changes to cell membrane composition, including increasing saturated fatty acids for stability at higher temperatures, upregulation of heat shock proteins, and alterations to key metabolic pathways. Perhaps most concerning is the documented ability of some species to horizontally transfer thermotolerance genes between distantly related fungi, further accelerating adaptation across the fungal kingdom. This genetic plasticity allows fungi to rapidly evolve in response to environmental pressures in ways that more complex organisms cannot match. Perhaps the most alarming example of fungal thermal adaptation is Candida auris, a multidrug-resistant fungal pathogen first identified in Japan in 2009 and now present in over 40 countries. Previously unknown to science, C. auris emerged simultaneously on 3 continents with no apparent connection, suggesting independent adaptation events rather than simple geographic spread. What makes C. auris particularly concerning is its combination of thermotolerance, surviving comfortably at human body temperature, antifungal resistance with some strains resisting all major classes of antifungals, and ability to persist on hospital surfaces for weeks. A study published in MBio in March 2023 found that C. auris isolates collected in 2022 demonstrate higher thermotolerance than isolates from 2018, suggesting ongoing adaptation to even higher temperatures. Hospital outbreaks have reported mortality rates between 30% and 60%, primarily affecting immunocompromised patients. Dr. Arturo Casadevall, chair of molecular microbiology at Johns Hopkins Bloomberg School of Public Health, has proposed that C. auris may represent the first example of a new fungal pathogen emerging specifically due to climate change-driven thermal adaptation. Genomic analysis of Candida auris reveals that it likely evolved from environmental fungi that inhabited wetlands and may have adapted to higher temperatures as these ecosystems warmed. The fungus shows remarkable genomic plasticity, with rapid chromosomal rearrangements in response to environmental stressors. The Centers for Disease Control and Prevention has documented over 1,700 clinical cases in the United States alone since 2016, with a particular concentration in long-term care facilities. Most concerning is the fungus's ability to develop resistance during treatment. In one documented case at a Chicago hospital, a patient's auris infection developed resistance to echinocandins, the last line of defense, within just 22 days of treatment initiation. This rapid adaptive capability makes CRS a particularly formidable emerging pathogen and potentially a harbinger of other fungal threats to come. The sudden emergence of thermotolerant fungi creates a significant immunological challenge for humans and other mammals. Our immune systems have evolved in an environment where relatively few fungi could cause systemic infection. As a result, our antifungal immunity relies heavily on innate immune responses rather than the more sophisticated adaptive immune mechanisms we've developed against bacteria and viruses. Research from the University of Minnesota published in Cell Host and Microbe in January 2023 demonstrated that thermally adapted fungal strains often display altered cell wall compositions that reduce recognition by pattern recognition receptors like DECTIN1 and DECTIN2, key components of our antifungal defense system. This immunological evasion, combined with limited antifungal drug options, only 4 major classes exist compared to dozens for antibacterials, creates a perfect storm for potential outbreaks. The World Health Organization recognized this emerging threat by adding fungal pathogens to its priority pathogen list for the first time in 2022, with CRS designated as a critical priority pathogen alongside bacterial threats like carbapenem-resistant Acinetobacter baumannii. Immunologists at the Pasteur Institute have identified another concerning trend. Thermally adapted fungi often produce novel secondary metabolites not found in their lower temperature counterparts. These compounds can have immunosuppressive properties, potentially helping the fungi evade host defenses. Additionally, the stress of adapting to higher temperatures appears to select for fungal strains with enhanced ability to form biofilms, structured communities of microorganisms that protect from immune responses and antifungal medications. Dr. Kieran Marr at Johns Hopkins has documented that thermally adapted fungi show an enhanced ability to sequester essential minerals like iron and zinc from host tissues, further compromising immune function in infected individuals. This metabolic adaptation represents yet another way that thermal selection pressure creates more virulent fungal pathogens, effectively turning the stress of climate adaptation into new weapons against host defenses. Beyond human health, thermally adapted fungi threaten food security worldwide. Wheat blast fungus, Magnaporthe oryzae pathotype triticum, has recently expanded its range from South America to Bangladesh and Zambia as warming temperatures allow it to survive in previously inhospitable regions. A single wheat blast outbreak in Bangladesh in 2016 destroyed over 15,000 hectares of wheat, demonstrating the potential scale of such threats. The agricultural implications extend beyond direct crop losses. Thermally adapted fungi often produce different mycotoxins, fungal secondary metabolites toxic to humans and animals, than their lower temperature counterparts. Research at Wageningen University has found that Aspergillus flavus, which produces carcinogenic aflatoxins in crops, synthesizes more potent toxin variants when grown at elevated temperatures. This suggests that climate change may not only increase fungal contamination of food supplies but also make that contamination more dangerous, creating a double threat to global food security at a time when agricultural systems are already stressed by changing climate patterns. Forest ecosystems face particular vulnerability to thermally adapted fungi. The sudden oak death pathogen, Phytophthora ramorum, has shown enhanced virulence at higher temperatures, threatening keystone tree species throughout western North America. Similarly, Dutch elm disease fungus, Ophiostoma novo ulmi, has expanded its range northward as temperatures warm, affecting previously resistant elm populations in Scandinavia and northern Russia. The chytrid fungus Batrachochytrium dendrobatidis, responsible for devastating amphibian population declines globally, shows evidence of thermal adaptation with recent isolates capable of growing at temperatures up to 2 degrees Celsius higher than historical samples. This adaptation potentially expands their range into previously protected highland amphibian populations. A consortium of mycologists from 6 continents, writing in Nature Ecology and Evolution in February 2023, warned that fungal thermal adaptation represents an underappreciated consequence of climate change that could simultaneously trigger cascading effects through ecosystems, potentially accelerating biodiversity loss across multiple taxonomic groups. The scientific community has begun mobilizing resources to address the fungal adaptation challenge. The Fungal Thermal Adaptation Consortium, a global research network established in 2022, works to identify genetic markers of thermal adaptation in environmental fungi that might predict emerging pathogens before they affect human health or agriculture. Meanwhile, pharmaceutical companies have renewed interest in antifungal drug development after decades of relative neglect, with several novel compounds targeting fungal-specific cellular processes now in clinical trials. These efforts represent a race against time as the thermal barrier continues to erode. Researchers are developing new surveillance systems to monitor environmental fungi for signs of thermal adaptation, creating an early warning system for potential emerging pathogens. Advanced genomic tools are being deployed to understand the genetic basis of thermal adaptation, potentially identifying targets for intervention that could slow or prevent the emergence of new fungal threats. Healthcare systems worldwide face significant challenges in preparing for emerging fungal threats. Unlike bacteria, which can often be treated with a wide range of antibiotics, fungal infections present unique treatment challenges. Fungi are eukaryotic organisms like human cells, making it difficult to develop drugs that target fungi without harming human tissues. The limited arsenal of antifungal medications, combined with rising resistance, creates a precarious situation for medical providers. Hospitals are implementing new infection control protocols specifically designed to prevent the spread of resistant fungi like auris, including enhanced cleaning procedures, patient isolation, and surveillance cultures. Medical education is being updated to increase awareness of fungal pathogens among healthcare providers, many of whom have limited experience with serious fungal infections. Diagnostic capabilities are also being expanded, with new molecular and immunological tests being developed to rapidly identify fungal pathogens and their resistance profiles, allowing for more targeted and effective treatment approaches. Addressing the challenge of fungal thermal adaptation requires unprecedented global coordination across multiple sectors, from healthcare and agriculture to environmental management and climate policy. The One Health approach, which recognizes the interconnection between human, animal, and environmental health, provides a framework for this coordination. International organizations including the WHO, FAO, and OIE are developing integrated surveillance systems to monitor fungal adaptation across human, agricultural, and wildlife contexts. Policymakers are beginning to incorporate fungal risk assessment into climate change adaptation planning, recognizing that warming temperatures may bring new infectious disease challenges. However, significant gaps remain in global preparedness, particularly in low- and middle-income countries where surveillance capacity is limited and healthcare systems are already strained. Effective response will require not only scientific innovation but also political will and resource allocation to build resilient systems capable of detecting and responding to emerging fungal threats before they become global crises. As global temperatures continue to rise, the future landscape of fungal threats remains uncertain but concerning. Models suggest that for each degree Celsius of warming, hundreds of additional fungal species may adapt to survive at mammalian body temperatures. This could fundamentally alter the pathogen landscape humans and other mammals have evolved with for millions of years. The most alarming scenarios involve fungi that combine thermal adaptation with other virulence factors, creating novel pathogens with no established treatment protocols. However, the situation is not without hope. The same genomic tools that allow us to track fungal adaptation also enable unprecedented capabilities to develop targeted interventions. Advanced surveillance systems may provide early warning of emerging threats, allowing preventive measures before widespread outbreaks occur. New classes of antifungal compounds targeting unique fungal cellular processes are in development, potentially expanding treatment options. The fungal thermal adaptation crisis ultimately represents both a profound challenge and an opportunity to rethink our approach to environmental change and infectious disease in an interconnected world. The thermal barrier that has protected mammals from fungal infections for millions of years is eroding as climate change drives unprecedented adaptation in the fungal kingdom. From the emergence of Candida auris in healthcare settings to expanding agricultural pathogens threatening food security and ecosystem disruptors compromising biodiversity, we face a multifaceted challenge that spans human health, agriculture, and environmental conservation. This phenomenon represents one of climate change's most overlooked yet potentially devastating consequences. As we've explored, Fungal thermal adaptation involves complex genetic mechanisms, immunological challenges, and ecological ripple effects that could fundamentally alter our relationship with the fungal kingdom. Our response requires not just scientific innovation but global coordination across disciplines and borders. The crumbling of this ancient thermal protection reminds us that even small changes in global temperature can trigger cascading biological adaptations with profound implications for planetary health. As we navigate this emerging frontier, our success will depend on recognizing the interconnectedness of human, animal, and environmental health in a warming world. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.